talk about new models for drug discovery.
Thanks. I'm really excited to be here. So we're a little biotech startup in Salt Lake City. Traveled down here because I was excited to meet with all of you and tell you about our story and learn from you and the questions that you ask. All right, so we're focused on rare disease. That makes sense. That's why we're here. What we're trying to do is sort of take, and I think you've seen this theme already today, drug discovery and biotech in general have been focused over the past few decades really sort of on single diseases, one disease at a time, and that makes sense. That's what the science enabled us to do. I think the trend you're seeing with Pearlstein Labs and with talks that other people have given, you'll see another one with Tooth Genomics in a little while, is to leverage technology to really try to accelerate drug discovery, to accelerate cures, and I think we're really on the cusp of a very exciting time in our history. So this is sort of the traditional biotech approach, and I'm going to talk through this real quick because it's important. This is a method that is pretty inefficient and pretty expensive, but it also has resulted in a lot of treatments for a lot of people. This is an effective method. It's just we're hoping that there are ways that we can make it more efficient. So basically, this whole process in traditional pharma starts with finding a target, and that comes from understanding the biology of a disease. So you spend years, or a lot of labs spend years studying a specific disease. You identify a target that you think that your team thinks is critical for that disease, and then you move through, and Ethan did a great job of talking about some of these things. You move through lead discovery, medicinal chemistry, in vitro studies, a number of different processes that take years to find one new chemical or several new chemicals that might affect this specific target. And then you make sure that it's not toxic, and then you put it in humans and you test for toxicity and you go through clinical trials. And at the end of the day, typically you've spent more than a decade and typically a very large amount of money, but perhaps you have a really exciting treatment. And one of the things I want you to keep in mind is that a lot of really good drugs don't make it all the way to the end. A lot of really good drugs fail somewhere in any of these regions. And so one of the strategies that we're using is drug repurposing, and I think most of you are familiar with that. But it is basically asking, can we take some of these drugs that have failed? Maybe they're safe, and maybe they were active against a specific target, but maybe that target didn't end up being as important in disease X or Y as was originally thought. It doesn't mean that couldn't be a useful treatment for some rare disease or some other disease. And so one of the things that we're trying to do, and I think others are trying to do, is leverage what's called drug repurposing or drug repositioning to cut out a number of these steps. The second thing that we're trying, and I apologize, with the widescreen, some of my slides are a little messed up, but the second thing that we're trying to do is parallelize this system. So if we're going to spend 5 years working on a disease, it seems like it would be better if we could spend the same 5 years working on 100 or 1,000 diseases in parallel. And I think you're starting to see people who are trying to figure out ways that you can do this. So I'm going to start with a story. This is the seed. This is what started this company, led me to actually drop out of medical school to pursue this. So I was doing my PhD in a laboratory at the University of Utah that had studied this particular disease, cerebral cavernous malformation, for more than a decade. So the laboratory had participated in identifying some of the genes that cause the disease, and they had made a lot of progress in understanding how the disease works. I don't think you need to be a physician or really know much about medicine to understand that this is an MRI you don't want to have. This is actually a patient from the University of Utah hospitals. You can see there are thousands of these vascular lesions all throughout their brain. So at the end of this decade, which is right about when I joined this laboratory, this was the state of the art. And I think for those of you who were at Brad Margus's talk this morning, you saw that giant complex map with ATM. I'm sort of preluding things here a little bit, but it's obviously got to be a lot more complex than this, or you might expect that it's likely to be much more complex than this. But after a decade with multiple labs working on this project, this is what people sort of understood about the disease. And so our lab in particular, we really felt, and we had, I think, some pretty good data to support that activation of this particular protein, it's not important which one, but that this portion of the disease was really what was driving the disease in these patients. And so we used a known drug that actually inhibits that pathway, and we hoped that it would make a difference. And we actually found, this is actually from mice, we built animal models of the disease, which is an important tool in many cases or most cases for drug discovery. We actually found that over time, the mice that got this repurposed treatment did not see any benefit. And that was incredibly disappointing. This was potentially a really quick way to affect patients. This is a drug that is relatively safe for most people. It's already in most pharmacies. A few small clinical trials, probably academic in origin, could have made a difference for these patients just using this drug, but it failed. And so that was a big gut check for us. But it sort of brought us back to this idea, especially in rare disease. There maybe are a handful of labs, maybe there's a handful of families who are doing their very best to understand a specific disease, but biology is really complicated. And that sort of is our core tenet. Biology is incredibly complicated and perhaps, in many cases, much more complicated than we give it credit for. And so this idea of sort of picking a specific target for certain genetic diseases is perhaps flawed. I know that it's worked, and there are great cases where picking a target, especially with like enzyme replacement therapies and other sorts of treatments, it's worked very well. But if you're looking broadly, if you want to try and fix 1,000 diseases, maybe this way is a little bit too slow. So one of the things that we noticed during this project was that the cells that had the disease looked really different. It's a really simple observation. These are normal human cells, the kind that line your blood vessels, and these are ones where we have modeled this disease. And all of us can see the difference. And I think this is powerful because there is a lot of information in structure, in images. People say a picture is worth 1,000 words, and I think it's true. There is a ton of information in this image. And what we realized was that if you could use and leverage technology to actually extract all of this information. I mean, think about when who's on Facebook? Everybody, I'm guessing, pretty close. When you post on Facebook, they know ahead of time, you'll see this like, oh, tag your friend, and it already knows your friend's name. They know that because they can use the structure of that person's face in the image, compare it against a database, and using facial recognition technology, know who that person is. Out of billions of people, out of more than 1 billion people, that same amount of information is in these images of cells, and it's incredibly complex. And so what we want to do is harness that. So we use pieces of the image, feed them into a computer, different aspects of the cells that we show the computer, and we let the computer learn what this disease looks like. And then the computer quantifies lots of things. So it figures out how big are the cells on average, how round are the cells on average. Are they sort of more oval or more star-shaped? We actually look at hundreds of different features, and this gets back, I think, to some of the things that Ethan talked about earlier, which was fantastic. This resonates with us in that biology is a black box still. We're learning more, but as much as we think we know, it's still a black box. And so what we're trying to do is embrace that. So for example, I may not know why the cell is star-shaped with this disease. I don't care. I just want to find a drug that fixes that, and then we can figure it out. That's sort of what we're trying to do. So we take all of these features, hundreds of features, we feed them into a computer, and the computer can come back and look at new images of cells, and it can do a really good job of saying, these cells have the disease, and these cells don't. That's a pretty good separation. And then we can add drugs, and again, I've already mentioned that we're really focused on repurposing and repositioning known drugs because we can cut through some of that long pathway of developing a new drug. So we add lots of known drugs, and we ask, are there any known drugs that when you add them to the disease cells, they can trick the computer into thinking that that disease cell is normal again? And if that happens, that is a great place to start to sort of look at the biology and figure out whether or not you have a treatment. So we did this for this particular disease. We identified 2 drugs that seemed in the cells particularly to really fix the disease. So one is a drug called Tempol. It's a tool compound people have used for years. It's not on the market. The other is vitamin D. And I think that's a really important, I mean, vitamin D is in the news all the time, but out of this blinded, unbiased system, vitamin D, which you can just buy at Costco for almost nothing, came out as a potential treatment for this disease. So we put them into our animal model, and you'll actually see here, well, it's a little bit hard to see, but all these little red dots are lesions in one of the animals that did not get the vitamin D treatment. You'll see the animals that got the vitamin D treatment had a little less than half the number of lesions. And so we published this. Another laboratory has published now with this information that patients with this disease tend to have strokes in February and March. So there's a lot of reasons why that might be. Perhaps it's associated with flu season. That also happens to be when your vitamin D levels are the lowest. And so perhaps we've stumbled upon a biomarker. Perhaps we've stumbled on potentially a treatment for the disease. There are a number of clinical trials, both sort of retrospective patient analysis that will be published shortly, and then a couple of prospective patient treatment studies that will be moving forward for this disease. So that was pretty easy. We'll see what happens, but that was a couple of years, and I think we went pretty quickly and probably got pretty lucky. But the next question we asked was, can we scale this? So this is a list of 2,000 diseases, each of which, and Ethan already introduced this very well, each of these diseases is associated with a specific gene. So these are monogenic diseases. And the question was, since we're already using robots to do a lot of our experiments, and we're using computers to do a lot of our analysis, why don't we just do the same thing for 2,000 diseases and see what happens? And so that didn't fly very well with sort of medical school and those sorts of things. So I dropped out, got some people to join me. We started a company to try and do exactly that. All right, how do we scale? And the rationale for using cell structure. So this is, does anyone know what this is? This is the subway map for Tokyo. But it looks a lot like the biology maps that we see. And so what I want to remind you is that look how much more complicated the subway map of Tokyo is than the image I showed you earlier about everything we knew about CCM. It's very, and I guarantee you the biology is much more complicated. If you were to see a delay at this train station, I'm not going to try to pronounce that, but let's say you saw a delay at this train station. One way to fix that might be to invest a tremendous amount of resources into understanding why the delay was happening at this train station. Another way to do it, if you could, would be to sort of install cameras at every train station all around the city and use algorithms to figure out when there's not a delay, what's happening, when there is a delay, what's happening. It's sort of a much more systems biology level approach, well, systems train level approach in that case. But we're trying to apply that same idea to biology. Let's be really agnostic about what we're looking at. Let's not try to outsmart biology because usually biology wins. So in biology, structure suits function. And this is the basis of phenotypic screening. This is the basis of pathology. This is the basis of all kinds of things. I don't think I probably have to prove this point to most of you, but the giraffe's neck is long so that it can eat the leaves. The neuron is long and branching so that it can make connections. And this patient in the upper right has Cornelia de Lange syndrome, and if you knew this disease and you knew these patients, you probably could almost make that diagnosis just by looking at the features of her face. It's very characteristic, and in fact, you'll find that for many diseases. And in fact, people have known this for a long time. Phenotypic screening, actually for identifying a new class of drugs using these sorts of things, like the cells are dead or alive, or the cells are bigger or smaller, that's called phenotypic screening. And it actually, compared to other types of screening, has been the best way to find new drugs. So people know this, but it's been a very analog sort of thing. People have been saying, for example, the cell is dead or alive. And in oncology, that makes a lot of sense. Perhaps when you're going into rare disease, there's a way to make it much more robust. So this is an illustration of that. This is like Google image result number 3 for phenotypic screen, maybe 4, I'm not sure. But what you'll notice down here is that there's red dots. You can't really see the blue dots, but there's red dots, less red dots, almost no red dots. This is the typical phenotypic screen, and it has been responsible for a large proportion of the drugs that are currently on the market. People just look for some change like this, like a decrease in the red. They've engineered this assay to specifically answer a question about one specific disease. Our thought was maybe there's a way that we could make an assay like this that's more complex, that works for every disease or most diseases, which seems pretty naive, but that's why we started a company, to go and try and do that. So let me see if this helps illustrate it a little bit. So I want you to imagine 2 features, and I've just picked these randomly. So one is the area of a cell's nucleus, which is that blue thing in the middle. It's sort of the heart of the cell, or the brain of the cell, depending on how you want to think about it. And another feature is the ovalness of a cell. So if I were to plot a whole bunch of cells from a whole bunch of diseases along these 2 axes, and then I were to plot cells from some other disease here, if you used just one of these, you would not be able to see a difference. But if you used both and you rotated your axes, you can actually see a nice separation between the 2 that would let you do a drug screen. And what we want to do is take this 1 to 2D and now move it to 1,000 dimensions, which is hard to draw, so I'm not going to try. So the first person we partnered with was Ann Carpenter at the Broad Institute. She pioneered a lot of the sort of things that we use, and specifically she's published this assay called the cell paint assay, where you can stain cells with these special markers that you can see. It's not showing up really well, but you stain cells, and with this pretty simple assay, you can see lots of different pieces of the cell. So you can see the nucleus, you can see the actin stress fibers, you can see the mitochondria, you can see a lot of the things that you've heard people talk about over the course of this conference as being involved in this disease or that. You can see them all in one assay. And then what you can do, so these are our images. We take these massive images of hundreds of thousands of cells. We model hundreds or thousands of different genetic diseases. We do that in as many cell types as we can. We're only at 4 right now, but we're trying to get to 12. And you start to ask for each of one of these, you start to measure more than 1,000 features. So we're not just asking how big or round is a cell, we're also asking, are the mitochondria close to the Golgi, or are they closer to the ER? We're asking as many questions as we can. We don't care why they're important. All we care about is identifying specific structural signatures associated with a specific disease. And so what I'm showing you here, here's healthy, these happen to be human epithelial cells, the sort of cells that you'd find on the inside of your lungs. You'll see in this particular disease, this actually happens to be Cornelia de Lange syndrome, you'll see that they look pretty different, and that's recognizable to you and I. This is another disease, you can see they also look different. That's recognizable to you and I. And for a small but important proportion of the cells, we see these differences. Now, biology is a little bit complicated. What I'm showing you here are columns. Each of these columns is a different feature of the cell. So the first one is the number of cells, right? So you might have a disease that kills the cells, and then that column is really low. So things that are high are in red, and things that are low are in blue. And what you'll see, we actually make, so for disease Z, for example, we actually make, you'll see 6 rows. So because the tools we use in biology aren't perfect, although there have been some recent ones that Ethan talked about with CRISPR that make them a lot better, but because the tools we currently use aren't perfect, we actually make multiple models of each of these diseases. We use robots to help us do that quickly, and we ask, are there features that are changing in the same way across all or most of the models? So you can see in yellow here, I've illustrated features for disease A where they're all red, they're all blue, and we use computers to help us extract that information. And so for this particular disease, we are pretty confident statistically. We can be as confident or not confident as we want, but we can quantify exactly how confident we are that each one of these features is specific to that disease. And so instead of having an assay where we're just looking at the shape or size of the cell or a decrease in red, we have an assay where there might be 48 different features or 150 different features that are all changing a little bit. And in fact, the computational power is such that we have statistically significant, repeatable, very robust phenotypes that we can use for drug screens that you or I can't see. And in fact, not even the best sort of microscope viewer pathologist, nobody can see them. They're just too subtle for the human brain to pick out. But by using computers, we can actually leverage the power and identify these phenotypes that we can use for drug screens. So what does that really look like? So we were enabled by a grant from NCATS. I know there was someone from NCATS in here earlier. I don't see him. But we were enabled last March by a large grant from NCATS at the NIH to really start moving pretty quickly on this. And since then, we've modeled more than 120 different genetic diseases. And I will say, just like what Ethan mentioned, we've really focused also on loss of function genetic diseases to start. The tools are just easier for us right now, but we are moving sort of our platform towards being able to do gain of function. And in fact, we have pilots working on infectious disease and all sorts of other, these images. This assay works for any biological perturbation. You can sprinkle anything you want and you see changes in the cells. So what you see here, these are 3 different cell types. Up is good. Up means confidence. And so what you see for 120 different genes, you'll see there's a pretty small subset of these, maybe 5 or 10%, where we feel like we have something that we can be really confident in taking forward. But what you'll also notice, let's look at like this green bar here. The green bar in these epithelial cells is really low. It turns out that gene isn't even expressed in those cells, so that's what we'd expect. But you'll see that the green bar in this other cell type is much higher. And so by looking at a lot of different cell types and by looking at thousands of different genes, our expectation is that as we go through these 2,000 genes that are sort of on our list right now, we'll be able to develop screenable models for hundreds of genetic diseases. And then we have to do the screens. So instead of focusing on a larger library, which is a very valid and useful way to go, we're focusing on a smaller library of drugs that, as I mentioned before, have sort of some evidence behind them for being safe or whatever. So these are repurposable drugs. So we take those drugs and we add them, and what I'm showing you here are all of the things that I circled in yellow before, sort of the disease-specific effects. All of those are shown in this axis. And you can see, for example, here are 6 different models of this hematologic disease. And here are 320 other models of more than 50 other diseases. And so when you're looking at more than 1,000 features, you can get this nice separation between the features. And I think in reality, this is something like 30 or 40 features that sort of make up this separation. Everything else, all the other effects, all the side effects are sort of in the y-axis here. So what we're doing now, just like I showed you before when we're trying to find a drug that fixes the phenotype, now we're trying to find a drug that does the same thing. Can we find a drug that we can add to one of these models that moves it back towards other diseases or sort of healthy? And that's a subtle but important distinction about why we consider healthy to be at the center of other diseases. I'd be happy to talk with any of you about that afterwards. We've done a lot of arguing about that point. So here's what it looks like. So I've reduced the number of controls here just to kind of show you, but we have sort of this origin of this is what healthy is, and then we have all of these models of the disease, and we add thousands of known drugs to those, and they look like this. And the size of the dot tells us something, and the darkness of the dot tells us something about how reliable in replicate that particular drug is. And what we're looking for are the set of drugs down here in this corner, which are moving us back towards the direction of healthy. Now, it's actually really hard to perfectly rescue 48 features or 56 features all the way back to healthy. So we're not looking necessarily for, I mean, it would be great if we saw a dot right over here on the left, but to us, statistically, seeing even a little bit of movement here, we have some confidence that that is a drug that it's important for us to sort of take into a set of validation. So again, what I've been trying to tell you about today is the way that we are trying to find shortcuts in terms of getting drugs to patients more quickly. And I think the theme of the conference, and I think the theme that you've been seeing across this entire industry over the past couple of years, is that a lot of people have a lot of really innovative ways to do that. Our sort of two main ways to do it, again, are to try and cut out pieces of this, cut out steps by repurposing drugs that we already know a lot about. There are scientists that spent entire careers working on a small number of drugs. We know so much about them, and it's a shame if they can't ever be used. And then the other thing we're trying to do is leverage technology and sort of our image-based assay to parallelize this process so that we can look at lots of things at once. So with that, I just want to tell you a little bit about the team and our funding. So when I started the company, I went and grabbed these two people. Was that like a 5-minute warning? Okay. Under pressure. Okay, got plenty of time. I'm hoping there's lots of questions. So this was my dissertation advisor, Dean Lee. So he's an MD, PhD cardiologist. He started 2 traditional pharma companies that are in clinical trials now, and he has a lot of experience with how things have typically been done, but he's been very open to sort of this new way of doing things. And this is Blake. I went to college with Blake. He is finishing his PhD in bioinformatics right now. He got that after he built an e-commerce company that he sold, and so he has a lot of sort of business expertise. We have a number of advisors, and I'll point out 2 or 3 specifically. This is Ann Carpenter at the Broad Institute, who sort of made a lot of this possible by the sorts of things that she published. And I can't tell you how fantastic, I mean, we all complain about the publishing and all those sorts of things, but all of the work that fantastic researchers do in academic institutions that our tax money and other things go to fund, they publish things that enable so much other great work to happen, and Anne is a great example of that. I think her paper has been cited something like 5,000 times, and I believe that there are 6 drugs in clinical trials right now, or about to be in clinical trials, that have come out of the software that she made with 1 NIH grant. So it's just fantastic to see those sorts of tools, and it's great to have her on our team as an advisor. Kirk Thomas made the first knockout mouse with Mario Capecchi at the University of Utah. He understands genetics better than almost anyone, and so he's a great person for us to have on the team because, as you can imagine, once we find a potential treatment, we now have to move it into an animal model to sort of validate that before we can take it forward to partners. And Perry Fell was the founder of Seattle Genetics, he was a scientist who became a founder who built a company that did very well based on a platform technology. And so we brought him in because we honestly have no idea what we're doing. So we've brought him in to help sort of guide us through the business side of things. We have a number of fantastic scientists, all PhD trained molecular biology and genetics students who have graduated and come to join our lab, a number of computational people. We were able to steal this one from 23andMe, which is fantastic, because when you're in Salt Lake and you steal someone from Silicon Valley, you feel like really excited. And I also want to just mention our funding. So there are these things called Small Business Innovative Research Grants. We have 3 of those. The first one was $1.5 million from NCATS. We got that in March. These 2 others, the NICHD, is the Eunice Kennedy Shriver Institute for Childhood Development. We got a grant from them to work specifically on a hit we already have for Cornelia de Lange syndrome, and the NHLBI grant is to work on a hit we have for Diamond Black Fan anemia. And those diseases just arose out of the data. We didn't really pick them. They were part of the original 2000 genes, but we had good data. They kept rising to the top. We had drugs that seemed to work, and so we've been able to leverage that into grants. We also have funding from the Rett Syndrome Research Trust, AT Children's Project. You may have noticed a couple of the images in my slides were in Brad's talk earlier, Cure HHT, and then of private investors. And so with that, I hope you guys have a whole bunch of questions. Thank you.
Thank you. Question?
That's never a good sign.
Question?
I'm sorry if you covered this because I came in a bit late. I know you started off talking about how you're repurposing failed drugs,
Yeah.
and I might have missed it, but is it difficult for you to get access to those failed drugs? Usually, this current system doesn't really incentivize publishing of failed results.
Yes.
So how do you do it?
It's really hard. That's a great question. It's really hard to get access to those drugs. So we are in negotiations with several large pharmaceutical companies who have libraries of those sorts of drugs. In the meantime, we're doing two things. So one is we have a library of drugs that we can just buy, and they are FDA-approved drugs and other sort of tool compounds that you can just sort of buy these libraries. There's two ways we can leverage that. One is when we find something, it's okay that it doesn't have a perfect business trajectory. We will publish that, and that will be proof of our model, and we will always do that. So we will always screen all of those drugs. Even if we get access to the large pharma drugs, we'll always keep this larger library of known drugs sort of in our pocket so that we can try and just get as many cures out the door or treatments as we can. The other way, and I notice that Max is sitting right behind you, the other way is, so there's a piece of legislation called the Open Act, which is part of 21st Century Cures. Am I getting this right so far? Okay. And what it does is it tries to incentivize exactly this sort of thing. So it says if there's a drug on the market and you're able to identify a new use for that drug and it's a rare disease, that you'll get an extra 6 months of patent protection, basically. I don't think it's exactly patent protection, but you get 6 months more of market exclusivity. So let's say that you are big pharma company A and you're selling a drug for $1 billion a year to 100,000 patients, and we come along and say, we found a new use for this drug in a disease that affects 500 patients. They're actually really incentivized right now not to move forward on that. Because what happens if we do a clinical trial and something bad happens? It might affect the sales of their $1 billion drug, right? So they're very scared about moving forward typically. But if this legislation passes, they're thinking, wow, pretty cheaply we could get this drug to the market potentially. We believe the science, it seems compelling. And if that happened, we'd get 6 more months of $1 billion a year in sales for our big indication, and we've helped these 500 rare disease patients. And that's a pretty compelling model, I think, from multiple sort of sides. I'm sure that there are issues with it, but it's something we believe strongly in, and there's an expert in the room on it. If you want to raise your hand, Max.
So yeah, we'll be talking more about this tomorrow, but you did a really nice job, Chris, laying that all out. But I want to add in that we're looking at the Open Act from kind of a 20,000 foot level, and we view this as something that actually has the potential to double the number of rare disease treatments. So basically, as Chris pointed out, there's really no good incentives for companies to be doing work in this space right now, which is hugely problematic for folks like you who just can't afford to wait any longer for treatments. So what we're asking people to do tomorrow, and this is like a little sneak preview for tomorrow morning, but we'll be having, hopefully all of you guys help us out and help yourselves out, call the Senate to remind them that incentives are needed to really push drug development forward. So we have that going on tomorrow morning, and then in the afternoon, Lisa and I will be doing a panel on 21st Century Cures if you guys want to learn more about that.
Thank you. That was not planned. That just naturally happened. Other questions? Yeah.
What about a common drug in Canada that's approved in Canada or Europe but not approved in the US? Do that pretty much with Gaucher disease, we know of a drug that can help type 2/3 Gaucher disease and cross the blood-brain barrier, but the drug's not approved in this country. Right. Can you use a drug like that?
Absolutely. And so we do our best to get all those sorts of drugs into our library. It's a little bit hard. What we're trying to not do is be a company that spends a lot of its time on any one disease or any one set of drugs or investing a lot of time or resources to getting that last long tail of drugs. But when we can find drugs like that easily and add them to the library, we do. What we're trying to do is not be the best at any one disease, but be 80% as good as anyone at most diseases. And so it's a little bit hard for us in terms of, it would take a lot of manpower to go look for every example like that and make sure we have it. But when it's easy to find, it's probably already in our library, and I could even go check for that specific case and see. And if it's not, it's easy to add. It's just one little pipette. So that's one thing I'll mention. There's a lot of genes and diseases and not very good databases that sort of make accessing all of this information easy. So we love hearing from patient groups who can tell us more about their disease. We love hearing about drugs that maybe we should add to our library. All of that is information that we sort of put into our system to try and make, all it takes is one drug and one disease to match. And so if we can add to either of those categories, it's really helpful. I think there's one in the, yeah.
Do you have a list of the diseases that you're currently studying?
Yes, we do. Let me go to it real quick. I know that was that was going to be the punchline. Yeah, so this is the list is here. There's just under 2,000 of them. We know that some of the diseases on this list probably aren't perfect. We had a computer generate this list using a number of factors. So we know that there are things on this that probably won't work in our platform, and we know that we're missing things. So I'd love for people to tell me more about the disease that they care about, and we'll add it. We met the mites, Matt and Christina Mite and Engelai 1, and so we added that to our list because we didn't know about it. It had not made it past sort of the computer cutoff to be on our list, but when we met that family, it was easy to add it in.
How do we get that disease on your list if it's not there?
So you can email us, you can talk to me after. Yeah, but basically the best we've had so far is just families that have emailed us, and then we sort of quickly read about it and throw it on the list if it looks like it might work. We have the list of things that might work really immediately, and we have NIH funding to support us looking at all of those. And then we have another list of things. I just met someone today who emailed us maybe a year ago or so, and unfortunately that disease isn't perfect for our platform right now, but we have it on another list for sort of as we expand the platform, sort of the next set of things that we can look at.
I was wondering, as kind of a side effect of what you're doing, if you're actually learning more about healthy cells and adding to the data on what a healthy cell looks like.
Yeah, that's a very important question, and it foreshadows what our real goal is. And so a lot of people think this is relatively naive, but I will say it anyway because I think you all are a pretty progressive bunch. So if we don't want to just look at these 2,000 genes, we want to look at the effect of every gene in dozens of cell types on thousands of morphological features, and the effect of every drug we can get our hands on all of those same features. And if you harness the power of computation correctly, you can actually start to maybe make that black box a bit more transparent. We can learn a lot about how different pathways are interacting, and this is sort of like what Facebook is doing with all of your images. They're learning a lot about you, what you like, what you don't like, where you might shop, all of those sorts of things based on what's in your Facebook profile. We're basically trying to make Facebook profiles for every gene. That didn't really come out right, but I think you know what I mean. And from that, start to really build a giant database of interactions. And I will say that other people have done this. So a great example is the connectivity map that the Broad Institute did. They looked at gene expression data for a lot of different perturbations, and people have sort of tried to fit. So if the gene expression data from disease A looks like this, and the gene expression data from drug Y looks like this, maybe you can put those together, and there's a few successes that have come out of that. However, gene expression data is still too expensive for them to look at every one of those interactions. Imaging data is not that expensive. So we can actually, we don't have the resources right now to look at all of those interactions. We have to be more selective. But it is not hundreds of millions or billions of dollars to look at every interaction of every gene with every other gene and every drug that we can get our hands on. Ethan, yeah.
Two questions, I guess, on the science. So one, it seems like I inferred that you're using like immortalized cell lines in this work. Are there plans to use primary cells or anything related to induced pluripotent stem cells or even co-cultures, I guess, since you're just looking at morphology anyway? And the second question has to do with how you create the model. So it looks like you're knocking things down with siRNA.
Yeah.
Are there plans, and it looks like you probably have multiple siRNAs per gene, so you make sure they all are consonant. Are there plans to, for example, compare to CRISPR knockout, or even a plan to shift from siRNA to CRISPR, or you think siRNA has legs for the long run?
Yeah, good question. So I'll address the second one first, which is we're working on that right now. I think CRISPR potentially does have the legs to take over siRNA. In fact, I just came from an siRNA conference. It's pretty clear CRISPR is a lot better. However, there are some technical hurdles that are associated with CRISPR right now that make it hard to look at thousands of genes really quickly. I mean, we've worked really closely with the Broad, and we know that right now, the biggest arrayed screen they've done, just to get into some tech talk, is 200 genes. And we want to do 2,000. And so in a year, probably it'll be like that. Right now, that would be $1 million. And so we're sort of just waiting for that perfect moment to jump in. We've got head-to-head comparisons of a bunch of technologies for 50 genes that we'll have in 6 weeks. And fingers crossed that CRISPR is as good for us as we expect. I know one concern, and I've talked to some scientists working in that space, is that sometimes if you totally knock out a gene, and you guys know this better than us, you end up just with death as the phenotype, and that doesn't work as well for us. So that's one thing we have to sort of toggle. Oh, yes. Sorry, the first question was, yeah. Yeah, so we actually do both immortalized and primary cells. I haven't shown all the data. There are certain issues with both, and so we're trying to balance both of those things. Right now we have 1 primary cell type and 3 immortalized cell types of different tissues, and as we grow to more than a dozen cell types over the next year or so, we expect a minority, but more than one of those to be primary cells. They're just harder to do the biology on. One of the things we actually talked about at lunch, though, is one of the nice things about this platform, and it echoes what Ethan's doing as well, when you look at multiple models, so if we find a phenotype in cell type A, B, and C, and a drug works for all 3 of those, you can imagine that's a pretty convincing piece of data for us to feel like we should prioritize that drug to move forward. But yeah, that's a great question.
Yep, thanks. So my main interest is mitochondrial medicine. So, part of what baffles me is the fact that Parkinson's and MS have been demonstrated to be the result of their dysfunction. However, the treatments for this disease do not treat the dysfunction itself. So, and we were more interested in mitochondrial disease, you could treat everything. So then I'm sort of, I don't know, somewhat fantasizing, I guess, or one can imagine, right, that Lipitor could treat everything. I mean, it's the best-selling cholesterol medicine.
Yeah, I will say, if you go to sort of drug repurposing conferences, just as a data point actually in support of that, what maybe sounds like a crazy idea, simvastatin is always a hit in everybody's screen. So probably we should all be taking it. I don't know, it seems to cure everything. No, but I mean, there are certainly core physiological pathways that probably play really important roles in a lot of diseases. And so one of the things that we might find is maybe there is a drug that we find works for several rare diseases, and that would be fantastic. I think it's probably unlikely that our technology would unleash all of those fantasies that you have, but we're going to do our best to rigorously test the hypothesis.
Optimism can take you anywhere.
Exactly.
Any more questions? We have time for one more. No? Okay. Well,
Thank
thank you so much.
Appreciate
Chris.
it.
We really appreciate it.